Okay, welcome back to Transcended Institute. So in this tutorial we are going to cover the topic which is regulation of gene expression and the concept of the Lac Operon. By the end of this topic, you should understand regulation of gene expression as well as the Lac Operon. Okay, so what you can see here, the diagram which I have here, this is in general what we are going to cover. So we're going to cover or to discuss whatever we can see on this structure. So all in all, we first have to start with regulation of gene expression. What do we mean when we say regulation of gene expression? After that, then we can now talk of the concept of the Lac Operon. And the diagram which we have here is nothing but the Lac Operon. So let's pause first for now for the Lac Operon. Let's talk of the regulation of gene expression. So regulation of gene expression, this is just the ability of the cell to control or to regulate what proteins it makes from its DNA. So we are saying that regulation of gene expression, this is just the ability of the cell to control. In other words, regulate simply means to control the proteins that it produces from its DNA. Proteins produced from its DNA. So that is what we mean when we say regulation of gene expression. As we know that all genetic materials are inherited in form of proteins, and these proteins will always come from DNA. So the cell has got an ability to control how these different proteins which we have in our bodies and in all living organisms can be expressed. We have different types of proteins. Some of the proteins in the bodies have to be expressed or produced on a daily basis. Then some of the proteins have to be produced maybe after some time. So how is the body of a cell able to control such? How is it able to control that some of these proteins have to be produced on daily basis and maybe in large quantities, and some of these proteins are produced on not on regular basis and in small quantities? How is the body able to control that? So that is what we call regulation of what? Regulation of gene expression. A good example can be red blood cells. We know that red blood cells, the RBCs, as an example of one of the proteins. So RBCs, we know that these have got a lifespan of what? Of about 120 days. So after 120 days, these RBCs will be distracted. So when they are distracted, what next now? They still need to replace these distracted what? Red blood cells. So the cell now, it means that the cell has to synthesize these other red blood cells which can replace those what? Distracted red blood cells. So how is the cell able to control such? That is what we call regulation of gene expression. So this regulation of gene expression, we are going to cover it much with the concept of prokaryotic cells and not in eukaryotic cells. So here we are just going to cover how are prokaryotic cells. By prokaryotic cells, we know that these are what? We are talking of bacteria. Prokaryotic cells, we are talking of what? Bacteria. So this is what we want to cover. How are bacteria able to what? To control the expression of genes. Okay. So here, the concept here which you have to have for you to understand this topic should be the concept of what? Transcription. We have covered transcription in prokaryotic cells, and we know what happens in prokaryotic cells. So if you have you understand transcription, then we are not going to have any challenges for us to understand this topic, which we call regulation of gene expression. So in other ways, the topic that we're going to cover is the topic which tackles transcription in prokaryotic cells. Okay, so as we know that all genetic material comes from DNA, and they have to be transcribed in from DNA, the central dogma of molecular biology, DNA, then we have RNA, then from RNA finally we have what? We have proteins. This is what we call the central dogma of molecular biology. So we know what happens here. From DNA to RNA there, we have what? We have the process of transcription. DNA has to be transcribed into RNA. Then from RNA now, it has to be translated to what? Proteins. So RNA to proteins, we have what? We have translation here. We have translation here. Then finally now we are going to have this protein, which is making the life of a living organism that simple. Okay, so we are saying the concept here is just mainly focused on transcription, but with transcription in a prokaryotic cell. So first we have to understand what are prokaryotic cells. We know that prokaryotic cells do not have a well-bound nucleus. So this DNA, which is going to give us a protein by the end of the day, is just found where? In the cytoplasm of prokaryotic cells. So meaning that in prokaryotic cells, transcription takes place at the same time as translation. What happens here is that if this DNA is transcribed, this messenger RNA, as we are going to have, messenger RNA is going to go straight to the what? To the ribosomes. So it's going to incorporate with the ribosomes. As they incorporate with the ribosomes, then this mRNA is now going to be what? To be translated into what? Into proteins. So these processes, transcription and translation in prokaryotic cells, takes place nearly at the same time. Immediately mRNA is produced, then translation also start. Then you finally have what? You finally have proteins. So you find that transcription in the prokaryotic cell, it's very simple. It's very simple for the fact that it doesn't involve those post-transcription modification which we do have in the eukaryotic cell, the splicing, capping, and polyadenylation. We don't have those here in transcription. And we are going to see the differences that this transcription in prokaryotic cell has over what? Over eukaryotic cell. So this is what we now call regulation of gene expression. How genes are able, how a bacteria is able to control the expression of what? Gene regulation. So just as I've already said, the concept here is going to tackle transcription in what? Or regulation of gene expression in prokaryotic cells. So the other thing which we have to know about prokaryotic cells is that genes on the prokaryotic cells, they are arranged in a manner that genes which perform similar functions, they arrange themselves or they group themselves next to each other, or they group themselves together on one side. Then the other set of genes which also perform similar functions will also group themselves on the other side. So genes in on prokaryotic DNA are grouped into two. So we have two groups of genes. These two groups of genes are what we call, we have one set of genes which is called structural genes. Then another set of genes is what we call regulatory genes. So regulatory genes. So these genes in a prokaryotic cell, they are grouped into two. So what happens is that these structural genes is going to be a number of genes, but these genes are performing what? Are performing similar functions. Then these regulatory genes, earlier on I said that regulate is to control. So these regulatory genes, these are just going to what? To control the expression of these structural genes. So we're going to have two sets of what? We're going to have two sets of genes on the DNA of what? In prokaryotic cells. So this is what we have to understand. Now, Such arrangement of genes where a particular set of genes which perform similar functions arrange themselves or group themselves together is what we call an operon. So I'm sure you have heard of operon. So that is what an operon means. Simply means a group of genes which perform similar functions arrange themselves together next to each other, one side or on the other side. So such is what we call an operon. So we have different types of operon. We can talk of the Lac Operon. We can talk of the Lac Operon. We can talk of tryptophan operon. We have different sets of these operons. But the operon, the first operon, was well understood in a bacteria. And the bacteria we know is what? It's a prokaryotic cell. So the bacteria, which these scientists who discovered an operon they used the bacteria and E. coli. And the first type of operon which they studied and which was to be first understood is the Lac Operon. It's the what? The Lac Operon. Lac simply means lactose. So that is just an abbreviation for what? It is an abbreviation for lactose. Lactose operon. So it was discovered that in a bacteria called an E. coli, the genes on the DNA are arranged in a manner that one set of genes is on the other side which perform similar functions. Then there is another set of genes on the other side which control the expression of these genes. And such is what we call an operon. Then this bacteria utilizes lactose as a source of energy. So that's why we have what? An operon. We have what? Lactose operon, which is abbreviated as what? As the Lac Operon. So at least by now we know the regulation of gene expression. Then now we have built this concept of what? We have built the concept of the Lac Operon. Then now we can go in detail to discuss much more in detail about this Lac Operon, which is in a bacteria called an E. coli. This is what now we want to understand. So from here now, then we can move on to understand the structure of what? Lac Operon. Okay. So when you see the Lac Operon, Lac here is just for what? For lactose. Then this operon is just a group of genes. This group of genes will be controlling the metabolism of what? Of lactose. Will be controlling the metabolism of lactose. So this is what we have. So now let's get to understand the structure of the Lac Operon. Okay. Here, this is what we have. The Lac Operon. Okay. We can. Okay. So let's have this structure and parts of the Lac Operon. So we want to see the structures and parts which we find on what? On the Lac Operon. Then later on we'll talk about the functions of each one and every part which we find at the Lac Operon. By the way, DNA in prokaryotic cell, as we know that it's circular, but for the sake of understanding this topic, we are not going to present this DNA in its circular form, but I will present it in its linear form so that we can understand better those parts. But this is what we know. The DNA is circular in a prokaryotic cell. So what we are going to have here, this is the DNA in what? This, we'll call this as a DNA in prokaryotic cell. So we are calling this now as the Lac Operon. Now this is what we call the Lac Operon. We have a group of genes grouped together performing similar functions. A clustered set of genes grouped together that perform similar functions. So by now we have already introduced that this DNA in the promoter in the prokaryotic cell here, it has got two parts of what? It has got two parts of genes. These two parts of genes are what we want to what we want to see here and understand where they are found and what's their main function. So this DNA of prokaryotic cell is going to contain what we call the promoter. As we have already understood the concept of transcription in a eukaryotic cell, so we had a promoter in eukaryotic cell. So we know the function of the promoter, but we are still going to talk about it. So in front of this promoter here, we are going to have a region which we call the operator. So what I've done here, P there is just for what? For promoter region. Then O there is what? For operator. So we have the promoter. In front of the promoter we have the operator. So P for promoter, then O there for what? For operator. These are the first sites that we'll find at on the DNA of what? Prokaryotic cell in the Lac Operon. Remember please, this is the Lac Operon and nothing else. Then in front of this operator now, we are going to have three different genes. So we're going to have these three different what? Genes. So these three different genes are given different names. So the first set of gene here, in front, just next to the operator there, is what is known as the Lac Z. Then next to Lac Z we have what? Lac Y. Then after Lac Y, finally we have Lac A. These Z, Y, and A, they are just letters of alphabet and do not present anything here. They do not have any meaning, but it's just to differentiate the different types of genes. So Lac here, as we already said, Lac for what? Lactose. So lactose Z, lactose Y, lactose C, A. Now these three are what we now call the what? Structural genes. Remember we just talked of what? We talked of the different two types of genes which we find at the Lac Operon. So the first set here is now out. We said we have what? Structural genes. So we have three structural genes at the Lac Operon. So I'll call these as what? Structural genes. So we have Lac Z, Lac Y, and Lac A. So these are the structural genes. And we said that structural genes, these are what are what? These are what code for proteins. So these genes here, which we call structural genes, they code for what? They code for proteins. In other terms, these are the genes which are encoded. So there is going to be transcription here. Transcription is going to take place. Then we are going to have mRNA. This messenger RNA is going to comprise the genes like Z, like Y, and like A. Then we said that this mRNA, it is in prokaryotic cell, it's going to incorporate with the ribosomes. As they incorporate with the ribosomes, then this mRNA is now going to be transcribed to give us those proteins. So those are the three structural genes which we find at the what? At the Lac Operon. Then later on we'll also talk of each and every protein which is encoded by each one of these genes. But for now, first let's understand the sites which we find at the promoter. So we are done with the right part here, the structural genes. Then this order, like Z, like Y, and like A, this order matters. So do not interchange these letters. They just like that, Z, Y, and A in front of operator there. Then let's come this side. We are the promoter here. So we'll now be moving in this direction to my left here. So next to the promoter, as we go to the left here, we are going to find a site which we call the cap site. So this region here, we'll call it the cap site. Please, this is a cap site. So cap there, this is just an abbreviation for what? Catabolite activator protein. So cap is for catabolite activator protein. activator protein. So here we are saying this is a capped site, meaning that this is not a cap but a site where this cap will bind. So later on, toward the end of this video, we are going to talk of this cap much more in detail, how it binds at the site there, at the cap site there. But for now, just know that next to the promoter, as you go to your left, you are going to find this region which we call the cap site, catabolite activator protein. Then next to the cap site here, we are going to find a gene which is known as, which we call Lac I. Lac I. This Lac I is for inhibitor. So I for inhibitor there. Inhibitor. So the purpose of this Lac I is it will just encode for an inhibitor. So we know that inhibitors are going to inhibit this process of what? Transcription. So once this inhibitor is encoded, it's going to inhibit this process of what? Transcription here. But that is the function, that is another set of genes which we find this side, Lac I, which encodes for an inhibitor. And the name of that inhibitor, we are going to introduce it later on. So the name of that inhibitor is what? It's the repressor. So we'll talk much more in detail what it all does. But for now, just know that Lac I encodes for an inhibitor which is known as the repressor there. Then now, each and every gene to be encoded, you need to have a promoter. The promoter is just the site where RNA polymerase binds, and as we know that RNA polymerase is the enzyme which transcribes the DNA for us to have messenger RNA. So you can't have transcription without RNA polymerase, and you can't have RNA polymerase without its binding site, which is the promoter. So if this is the gene which will be encoded, then behind it we are supposed to have what? We are supposed to have the promoter. This is not the same promoter which is going to control which is going to control the expression of the structural genes. This is the promoter which is just going to control the expression of Lac I gene here. So because of that, to differentiate it from this major promoter, we'll present this one as a promoter I, promoter prime there, or you can say promoter Lac I. You see, one and the same. Promoter Lac I or promoter prime. So this is what we'll find. Now, let's look at this. Starting from here all the way up to here, this region now is what we call, regulatory genes. Remember we talked about the two groups of genes which we find on what? On the Lac Operon. So this side is what we call regulatory genes. So now we have these two types of genes: structural genes, which we just talked about, structural genes, and regulatory genes. So at this point now we know the genes which we find on the structural genes, the parts, as well as those which we find are the regulatory genes. So these regulatory genes is just to control. These are controlling genes. So in other words, regulatory genes are called what? Controlling genes. Controlling genes. These genes, this side, are the ones which control the expression of what? These structural genes. For these to be transcribed, like Z, like Y, and like A, for them to be transcribed, it will be under the influence of regulatory genes. If these genes regulate for the transcription to go ahead, then these structural genes are going to be encoded. If not, then we are not going to have what? These structural genes are not going to be expressed. So that is just the function of regulatory genes. Whereas structural genes, those are encodes for protein. So this is the Lac Operon, Lactose Operon in a bacteria, E. coli. Now we understand the structure of the Lac Operon. What is remaining now is for us now to understand the functions of each and every each and every site which we have here at the Lac Operon. Okay, so before we talk of that, let me give you the primary idea behind the so-called Lac Operon. The primary idea behind the Lac Operon is just the principle based on a bacteria called what? E. coli. E. coli bacteria, this is found mostly in guts. In the gut, such as a small intestine. So that's where an E. coli is found. In the small intestine. Now, this bacteria E. coli, its survival depends on sugars. Its survival depends on sugars or carbohydrates. So we can talk of different types of sugars here. We can talk of glucose. We can talk of lactose. We can talk of galactose and others. Galactose. So the survival of this bacteria depends on what? These sugars. Now, the most preferred type of sugar that this bacteria likes to utilize is glucose. The reason being is because if we compare this glucose to galactose, or to lactose here, we are going to find that this glucose, it is the smallest, it is in its smallest form of a sugar. It is a monosaccharide. So glucose is a monosaccharide, whereas lactose, this is a disaccharide. Now, then this galactose, let's get rid of it, because we know that from lactose we can have galactose. So let's just talk of these two. So the principle behind the Lac Operon depends on the sugars. How this E. coli survives, its survival depends on sugars, and these sugars will depend on the host. A host, in this case, we are referring to you as a person. So the sugar that you take, those are what this bacteria is going to utilize for it to survive. So the simplest form, the preferred type of sugar is glucose. Then if glucose is not present, it will utilize what? Lactose. Lactose. The major source here is what? Milk. Major source of lactose is what? Milk. So if glucose is not present, meaning that now the bacteria is going to utilize what? Lactose. So this is where now I was saying the principal idea behind the Lac Operon depends. It depends on lactose. It depends on lactose. We are saying that lactose is a disaccharide which is made up of two sugars. It is made up of what? Two sugars. And what are these two sugars? Lactose is made up of glucose and what? Galactose. So for when glucose is not present in the medium, for a bacteria to survive, it's then going to utilize what? Lactose. So this lactose, a bacteria cannot utilize lactose in its what? Disaccharide form. It has to break down this lactose into its smallest unit, which is glucose and galactose. So the breaking down of lactose into smallest units, which is glucose and galactose, meaning that here there is need to break that what? Glycosidic bond. So we know that lactose and galactose, they're linked through what? A glycosidic bond. So this bond has to break. So the breaking down of this glycosidic bond to give us these smallest units of lactose, which is glucose and galactose, involves energy. So now, I think when I said that the most preferred type of sugar that a bacteria likes to utilize is glucose, you can now get that reason. So the reason to that is because if glucose is present, there will be no need for a bacteria to use energy, because glucose, it is in its what? Simplest form. Whereas if glucose is absent, there is only lactose in the medium, then the bacteria now have to use energy to break down this lactose into its smallest unit, which is glucose and galactose. Then now a bacteria can use this glucose, and this galactose is going to be modified by another enzymes within the cell. It is going to be modified back to what? To glucose. Then a bacteria will then also use what? This glucose. So that is this is the principle behind. Now we are talking here of breaking down lactose into smallest units. Then again, the breaking down of this glucose, or this lactose, sorry, involves what? Involves the energy. So there is need for enzymes to be encoded by a bacteria which can what? Which can break down lactose into smallest units. That is the whole reason why we have these structural genes. Well, these structural genes are the Lac Operon. We need these structural genes. Some of these genes which are encoded by these genes, we're going to see the functions. Some of them is just to break down what? The function of those proteins or genes is just to encode for a protein which can break down what? Lactose into its smallest unit. So that is the whole reason why we need these structural genes. So meaning that in the presence of glucose, when glucose is present, there is no need for these structural genes to be encoded. The reason why is because you don't need any enzyme to break down glucose. It is already the simplest form of a sugar. So there is no need for that enzyme. But in a case where you have lactose, that's when you need these structural genes to be encoded so that they can break down lactose into smallest unit. So such metabolism of lactose is what we call the Lac Operon in the bacteria. Okay, so that is the principle behind this Lac Operon. Then now we can talk about the function of or the type of proteins which are encoded by each one of these. Then we can also talk about the functions of those proteins. Then also the function of each and every site here. Later on, then we'll talk about how these regulatory genes are able to control what? The expression of these structural genes. So we are done with the parts of the Lac Operon. Then now let's talk of the proteins which are encoded by each and every structural genes here. So remember that we have 3 structural genes at the Lac Operon. So we have Lac Z, Lac Y, and Lac A. Then at the end there we're going to have terminator. So or termination site. So we know that that will be the site where the process of transcription will have to come to an end. That is very much similar with that in what? In eukaryotic cells. Okay. So now let's talk of the proteins which are encoded by each one of these structural genes which we have here. So Lac Z, what type of protein encodes for Lac Z? So let's have this one here. Okay, so Lac Z or Lac Operon is going to encode for a protein which is known as beta-galactosidase. So Lac Z, remember, Lac Z is going to encode for this protein which is known as beta-galactosidase. Please take note of that. Okay, then Lac Y. Lac Y is going to encode for a protein which is known as beta-galactosidase permease enzyme. As we know that proteins are enzymes, or enzymes are protein in nature, that's why here I'm interchanging the names. So protein or enzyme, I'm just talking of one and the same. So Lac Z encodes for beta-galactosidase, then Lac Y is going to encode for beta-galactosidase permease enzyme. Then finally we have Lac A at the end here. Lac A encodes for a protein, beta-galactosidase transacetase enzyme or protein. So these are the respective proteins that each one of these structural genes encodes. Lac Z, beta-galactosidase, then Lac Y, beta-galactosidase permease enzyme. Lac A, beta-galactosidase transacetase enzyme. Then in other books, you are going to find that this, like Y, they are going to denote that it encodes for permease enzyme. Then this, like A, encodes for what? Transacetate enzyme. It's just one and the same. It's like it's more like beta-glycosidase is a surname. So some of the books will not include the surname. They will just say permease enzyme or transacetate. So don't get confused that there it was beta-glycosidase, but how come here I have this? But it's just one and the same thing. Now, let's talk of the functions. The functions of each one of these proteins. Remember that when transcription takes place, these are what are going to be? These are all going to be on the same mRNA. When transcription takes place here, these structural genes are going to be transcribed on the same mRNA. Then here, let's say this is our mRNA. So this can be like Z, this one can be like Y, then here this one can be like A, all on the same mRNA. Then this mRNA now will interact with that, we incorporate with the ribosomes within this cytoplasm. When it incorporates, it's going to, the ribosomes are going to translate these proteins. Then when they translate, that's when we are going to have these three different proteins here, which we call the structural, which have been encoded from the structural genes. So this is the reason why I said that in prokaryotic cells, transcription and translation takes place at the same time, because immediately this one is transcribed, then translation also starts taking place. Then you have what? You have proteins there. Then these proteins now can be used for those different functions which we want to discuss just about now. Okay. So let's talk over the functions of each one of these. So let's start with a like Z, which encodes for beta-glycosidase. So what is the function of beta-glycosidase? Beta-glycosidase. So beta-glycosidase, which is encoded by a gene like Z, this one, its function is just to break down lactose into its smallest units. So its function is just it breaks breaks down lactose into its smallest unit. So the smallest units here, we know we are talking of glucose and galactose, because those are the monomers of what? Those are the monomers of lactose. So that is the function of beta-glycosidase. That's why I said that in the presence of lactose, that's when you need these structural genes to be transcribed, because there's need to break down what? Lactose. In the presence of glucose, you don't need these enzymes to be transcribed, because there's no need to break down what? Glucose. Okay. So we are done with the beta-glycosidase. Now, what of permease enzyme? Beta-glycosidase permease enzyme, which is encoded by what? Which is encoded by the gene like Y. What is the function of this enzyme? So the function of permease protein is just to allow, by the way, this permease enzyme, this is a membrane-bound protein. It is found in the cell membrane of the bacteria. So if let's say this is the bacteria, this is the cell membrane. This is the cell membrane of the bacteria. That's where this permease enzyme is found. That's why we are saying that it is a membrane-bound what? It is a membrane-bound protein. It is found in the cell membrane. So what is its function? Its function is to arouse lactose, allows lactose gets inside the cell. So let's say its function is to allow lactose to get inside the cell. Inside the cell. By the cell here, we mean the cell of the bacteria. So if this is the bacteria, this is going to be what? the inside of the cell of the bacteria. Remember, this is the cell membrane. Then outside here, this is now your gut. That's where your small intestine, these are your small intestine. That's where this lactose is going to be found. So there's need for this lactose to be pumped from what? From the guts here to the inside of what? To the inside of the bacteria there, to the inside of the cell of the bacteria, so that this bacteria now can utilize that lactose. So that is the function of what? Permease, just to allow this lactose get inside the cell of the bacteria. Nothing else. So that's it. So now you can see that permease is going to allow lactose get inside the bacteria. Then there's need now to break down that lactose. That's when now you need beta-galactosidase. So that's why, as we already talked about, that similar genes performing similar functions, they are grouped together. This is what we meant. So as we have seen, all of these are related to what? Lactose. Such is what we call an operon. Then finally, let's talk of transacetase, beta-galactosidase transacetase. A lot of books, or let me say some other books, are going to say that the function of transacetase is not well known, is not well understood. But there's still something that we can talk about it. So transacetase, this one is its function is it transfers an acetyl group to beta-galactosidase and activates it. Acetyl group to beta-galactosidase and activates it. Sidase and activates it. What we mean here is that this transacetase, that's why it's very easy to know the function of enzymes. Transacetase here, this trans is coming from transfer. So meaning that this type of protein, it is capable of transferring something. So what in this case, this transacetase, it transfers an acetyl group. It transfers an acetyl group to where? To beta-galactosidase. Then when this beta-galactosidase combines with this acetyl group, it becomes activated. Once it becomes activated, then now it breaks down lactose. It breaks down lactose into its smallest units. But just as I've already said, other books are going to say that the function of transacetase is not well understood. So that's it. Now we are done with these structural genes. We have talked of 3 different structural genes, what they encode for, then even their respective functions. So by now at least you can feel the feeling of Lac Operon. At least at this point you can answer some questions, some exam questions with regard to the Lac Operon. But there's still a lot to talk about. Let's now talk of the operator, then we talk of the operator, then promoter, capside, Lac I as well as Lac P. Okay. We are done with the structural genes. Maybe before I move on, let's first talk of one more important point here. If we look at these genes, Lac Z, Lac Y, and Lac A, these are going to be transcribed at the same time under the influence of one promoter. So this one promoter here, when RNA polymerase binds here, all of these genes, Lac Z, Lac Y, and Lac A, are going to be transcribed at the same time. So because these different genes are transcribed under the influence of one promoter, such type of transcription is what we call polycistronic. Polycistronic. Poly simply means many. Then cistron is another name for genes. Cistron is a Latin word for what? Genes. So when we say polycistron, what we mean is that many genes. These many genes are all transcribed under the influence of one promoter. So that is what happens in prokaryotic cell. So the transcription in prokaryotic cell is said to be polycistronic. Simply because different genes or many genes are transcribed under the influence of one promoter. This is a different story when it comes to eukaryotic cell. In eukaryotic cell, transcription is said to be monocistronic. Monocistronic. What it means there is that mono, we know that simply means one. So one gene. So each gene in eukaryotic cell has got its own promoter. That's why transcription in eukaryotic cell is said to be monocistronic. So if this was in eukaryotic cell, it would have been saying that this like Z was going to have its own promoter here, as well as like Y its own promoter, then like A also was going to have its own promoter. That is in eukaryotic cell. Monocistronic, eukaryotic cell, polycistronic, prokaryotic cells. Okay, so now we can move on to talk of these other sites which we have here. Okay. All right. So let's talk of the regulatory portion here. The regulatory portion here. So remember now, we said we have the promoter here. The function of the promoter, we said that is what? It's just the binding site for RNA polymerase enzyme. So with the promoter there, we are saying it's just what? The binding site for RNA polymerase. For RNA polymerase enzyme. So this, we know that we cannot have transcription without this enzyme, RNA polymerase. So RNA polymerase is going to bind where? At the promoter. Then it will start reading the DNA strand there, thereby transcribing these genes. So that's the function of the promoter. Then let's talk of the operator. What's the function of the operator? The function of the operator is also the binding site for an inhibitor. We talked of, so we are saying the operator, this is just what? The binding site for an inhibitor. For an inhibitor. So inhibitor is going to inhibit the process of what? Transcription. And we say that this inhibitor is what we call what? The repressor. So this repressor, which will act as an inhibitor of transcription here, is going to bind where? At the promoter. So ideally what will be happening here is that if this repressor binds at the operator there, then here, remember we have RNA polymerase. RNA polymerase is behind here at the promoter. So RNA polymerase, for it to transcribe these genes, it has to move in this direction, because that's where we have transcribing genes, structural genes. So it has to move in that direction. So now because we have that repressor there at the promoter, this RNA polymerase will not be able to move this in this direction. It will be blocked by the repressor. So that's why we call this one as what? Inhibitor. It's inhibiting the process of transcription. In that case, it means that these structural genes are not going to be what? These structural genes are not going to be transcribed. So we'll talk much more in detail later on, but ideally that is what happens. So now we have talked of the function of the promoter, the function of the operator. Then this cap site, we'll talk much more in detail later on, because it is one of the positive control mechanisms So talk about it. It is just a site where a catabolite activator protein will bind. So for now, let's reserve that. Let's now talk of Lac I. This Lac I, we say that it encodes for Lac I gene. It encodes for Lac I protein. We said I here, I for inhibitor. I for inhibitor. So this inhibitor is what we called repressor. It's what we call repressor. So by now I think you can relate. Here we have the repressor. We are just from mentioning that the repressor is an inhibitor, so it's going to inhibit the process by binding at where? At the promoter there. So I think by now you should be relating. So we have an inhibitor or repressor. This protein is encoded by a gene which is called Lac I. So that's the function of Lac I, just to encode for an inhibitor which is called the repressor. Then, for any gene to be transcribed, what do you need to have? You need to have a promoter. The function of the promoter, that's the binding site of what? The RNA polymerase. So transcription cannot take place, be it in eukaryotic cell and prokaryotic cell, without RNA polymerase. And this RNA polymerase cannot start from anywhere. It has to start from the promoter. That is its binding site. So behind each and every gene which you want to be transcribed, behind that gene there must be a promoter. So that is the reason why here, behind that Lac I gene, we have what? We have this promoter. So we have presented it as a P prime or initial, or some other books are going to present it as a P Lac I. So it's the promoter for Lac I. So for this gene to be expressed, you need to have this promoter. That's where RNA polymerase which will transcribe for this gene will bind. So at least now we have talked of the function of at least each and every site. Okay, so this is what happens. So the direction here, these, as we know that transcription always runs in that direction, 3' to 5'. So that's why we have those 5' here and 3' there. So this is what happens in the Lac Operon. Then now we can talk of the conditions which facilitates this process of transcription here. We can talk of how the levels of glucose and lactose affects the Lac Operon. Okay. So let's have the same. This is the same DNA which we have, the Lac Operon. So as, by now we know that at this Lac Operon we have the promoter, the opalator, then we have Lac Z, we have Lac Y, we also have what? Lac A. Then here we have the cap site. Then behind the cap site we have Lac I. Then behind the Lac I we have the promoter for Lac I there. So now what I want to talk of is the two conditions which facilitates the process, which affects or activates the process of transcription. This is transcription here in the Lac Operon is affected by what? By the levels of lactose and glucose. Lactose and glucose. So here now, that's where we can talk of the on and off theory of the Lac Operon. So there is what we call the on and off theory of the Lac Operon. This on and off theory of the Lac Operon depends on the concentrations of these two sugars here, which we have here. So lactose and glucose. So what happens? Remember, this is the bacteria which just depends on the diet that you take. If you take glucose, a lot of glucose, meaning that this bacteria is going to find it very easy to utilize what? Glucose. But what if you take glucose in a form of what? In a form of lactose. Let's say you are just from taking milk. The bacteria, meaning that in that case glucose levels will be low, or there will be no glucose at all. Then lactose or E. coli will not have any option other than to utilize lactose so that it can obtain what? It can obtain glucose from lactose. So this is what is going now to be the condition or the Lac Operon, to turn it on or off. So there is that switch, and that switch is on what? The on and off switch is on the operator. So this is what now we want to see. So let's start with if there is a high concentration of glucose in the medium. So let's say that glucose is present. High concentration of glucose is present, then lactose is absent in the medium. In this case, the medium is your guts, the small intestine, and all those other types of can talk of the GITs. That's where this bacteria is found. So here now we are saying if glucose is present and lactose is absent, what will happen to this Lac Operon? The Lac Operon in this case will be said to be off. But how? When glucose levels are high, the Lac Operon is said to be off. So how? This is how. If glucose is present, this LAC-I gene is going to be activated. Once LAC-I gene is activated, it's going to encode for that protein we said, LAC-I inhibitor repressor. So it's going to encode for active what? Active repressor, which is an inhibitor. So in the presence of high concentration of glucose, or even if the concentrations are low, but as long as glucose is present in the medium, this LAC-I gene is going to be activated. Once it's activated, it's going to encode for a protein LAC-I repressor. So this protein is going to be encoded. Once this protein is encoded, so let's say this is the protein that the repressor. Let's say this is the repressor. It has been encoded. So this repressor now, once it's encoded, it's going to bind where? At the operator. So you can now relate why we said the function of operator is the binding site for the repressor. So this is what happens. This will only be in a case where there is high concentration of what? There is high concentration of glucose in the medium. This repressor will be encoded by LAC-I gene. Then the repressor will go and bind where? Will go and bind at the operator. So now let's see what effect is this going to bring. Once this repressor binds at the operator there, RNA polymerase will still bind where? At the promoter there. Remember, transcription cannot take place without RNA polymerase. So let's say this is our RNA polymerase there. This is our RNA polymerase there. RNA polymerase, it is the enzyme which can only transcribe what? Which can only transcribe this DNA gene to messenger RNA. So what happens now here is that at the operator here, we have at the operator there we have the repressor. This repressor will bind very tight to the operator there. So when this messenger RNA, RNA polymerase would want to start transcribing this DNA, it will not be possible because it will be blocked by what? It will be blocked by this repressor here. As a result, these structure genes will not be encoded. So these structure genes will not be expressed. So because these genes will not be expressed, that's when we say that the Lac Operon is turned off. So when you have the repressor, the operator there, the Lac Operon is off. The reason being is because RNA polymerase is being blocked from transcribing the DNA strand. So this will only be in the situation where you have high concentration of what? High concentration of glucose in the medium. The story will not change. The story will be the same if, let's say, we have a condition where glucose is present, then let's say lactose is also present. What will happen in this case now? Will be will the Lac Operon be on or off? The Lac Operon in this case, when both lactose and glucose are present in the medium, the Lac Operon will still be off. Why the Lac Operon will be off? Because we said earlier on, we said that the most preferred type of sugar that the bacteria likes to utilize is what? It's glucose. So when lactose is present, glucose is also present. The bacteria will not go for lactose, because remember, for bacteria to utilize lactose, it has to break it down, and breaking down lactose involves the energy. So in that case, it will only go for what? It will go for glucose. As a result, when the bacteria is utilizing glucose, meaning that at the operator here, there is what? There's a repressor. Hence, these genes will not be encoded. Overall, the Lac Operon is off. So this is what happens. That is the condition. So as long as you have high levels of glucose, or you have glucose in the medium, just know that the Lac Operon is off. Okay, we have talked of this one condition. Now let's talk of if we have high concentration of lactose. So let's say now we have this condition. Lactose is present, then glucose is absent. Glucose is what? Glucose is absent. What happens now? Will the Lac Operon be said to be on or off? So in this case, when the Lac Operon, when there's a high concentration of lactose in the medium and glucose is not present at all, what will happen now is that this bacteria, remember we said that this is the bacteria, this bacteria has got a membrane-bound enzyme. We talked of beta-galactosidase permease enzyme, which is encoded by gene like Y. So what happens here is that this now is going to be what? Let's say these are your small intestine. This is the medium. So in the medium now here we have what? We have these, let's call these as what? Lactose. So these lactose here, they will have to be allowed or to be pumped where? Inside the cell of the bacteria. So the pumping of these lactose from the medium, from the surrounding medium to the inside of the bacteria cell, this needs that enzyme, permease enzyme. So the function of the permease enzyme, we say that is to allow these lactose to get where? Inside the cell. So this is what will happen in the presence of what? Lactose. Lactose is present. Then permease enzyme will allow that lactose get inside the cell of what? Or the bacteria. Then when it gets inside the cell of the bacteria here, we now know the story. This lactose has to be broken down. So the breaking down of that lactose needs another set of enzyme, which we just said is what? Beta-galactosidase enzyme, which is encoded by gene like Z. So this is now what will happen. So let's see how this Lac Operon is going to be turned on in the presence of lactose. When lactose is present, so we are now saying we have lactose in the medium. This lactose, when it is present, is going to act as an inducer. An inducer is a molecule which can activate or inhibit any process. So it's going to be an inducer. So in this case, lactose is going to be what? An active inducer, meaning that it's going to activate the process of transcription. So how is this lactose going to act as an active inducer? What will happen is that this lactose, when it is present in the medium, is going to be modified a bit to become what? Allolactose. When lactose is present in the medium, it will act as an active inducer, meaning that it's going to activate the process of what? Transcription here. So once lactose is present there, we are saying it's going to be modified a bit to become allolactose. The reason why it's going to be modified to become allolactose is because this lactose wants to attain the binding sites of what? the repressor. So in its normal form, in its natural form of lactose, this lactose cannot bind with what? With the repressor. The reason here is that this lactose, we are saying, is an inducer. What will happen is that lactose later on is going to bind with this repressor here, which is sitting on the operator. When they bind here, when lactose binds with that repressor, then this repressor is going to lose its binding affinity with the operator. So when it loses its binding affinity, meaning that the whole thing now it's going to move out of out of that it's going to move out of the operator. Then this polymerase now can read or transcribe these structural genes. So once lactose is modified a bit to lactose is going to gain the binding affinities of the repressor. It's going to gain the binding affinities of repressor. Then this now lactose will go and bind with what? With the repressor at the operator there. So when lactose binds with the operator there, this now repressor will lose its binding affinity with the promoter. Once it loses its binding affinity, it will have no any strength to continue sitting there or staying there. So it will move out of the operator. Once it moves out of the operator, meaning that now this RNA polymerase, which was being blocked by the repressor there, it can now find it easy to what? To read or transcribe this DNA. As a result, we are going to have what? These three different proteins. So that is overall is what will happen. So that's how this lactose is going to act as what? An inducer. Simply binding with what? With the repressor at the operator. When it binds with the repressor will lose its binding affinity, thereby moving out of the operator. When it moves out of the operator, then this RNA polymerase now can transcribe this DNA. As it is transcribing, then these three different genes are going to be encoded. They'll be encoded then in a form of mRNA. Then this mRNA will interact with the ribosomes, thereby producing the what? Producing the proteins which we need. Then at this point, it would mean that these structural genes have given us all those three different what? Three different proteins: beta-galactosidase, permeans, as well as transacetase. These now can work together to break down what? This lactose in its smallest unit, which is galactose and glucose. Then the bacteria can now utilize that glucose and it survives. So this is how the Lac Operon is turned on. So in this case, when lactose is present, the Lac Operon is said to be on, because these structural genes will be encoded. So that's the on and off theory of the Lac Operon. The conditions just depends on the concentrations of these two different sugars which we have here. So that's the Lac Operon. Okay. So just know that if you have both glucose and lactose in the medium, the Lac Operon will be off, because the bacteria will go for glucose, which does not need to be broken down. When lactose is present, glucose absent, the Lac Operon will be on, because there will be need for these structural genes to be transcribed. So that's the theory behind. Okay, then now finally we can talk of this capside, where we can now talk of the positive and negative control mechanism. Okay. Then we can claim that we are done with the Lac Operon after covering this. So let's talk of the negative and positive feedback of the Lac Operon. Let's start with the negative feedback mechanism of the Lac Operon. So negative feedback mechanism. The negative feedback mechanism of the Lac Operon. So want to see what molecules are going to inhibit the process of what transcription here in the Lac Operon. So those molecules which are capable to inhibit or slow down the process of transcription in the Lac Operon are what are regarded to be what negative feedback molecules. So this is also what we call negative control mechanism of the Lac Operon. So at this point, if you have been following well, you even know some of the molecules which will be regarded here as what negative feedback, which are controlling the negative feedback mechanism here. We can talk here of what? Or the repressor. Repressor is one of those negative feedback mechanisms. We know that when the repressor is present, it's going to bind where? At the promoter. Oh, at the operator, sorry. When it binds at the operator there, what will happen? There will be no transcription. These genes will not be transcribed. So because there's going to be no genes here which will be transcribed, that is what we call negative control. So repressor is that one example of negative controlling feedback mechanisms. Nothing much about the negative feedback. So this one is encoded by Lac I inhibitor. So the negative control mechanism inhibits the process of what? Inhibits the process of transcription here in the Lac Operon. And a good example there is what? It's the repressor. What of now the positive feedback? Positive feedback control mechanism. So the positive control, these now are the molecules which will be what? Will activate. To activate simply means to speed up or facilitate the ongoing of the process. So these we're going to have what? Activators. These will be molecules which will speed up the process of what? The process of transcription here in the Lac Operon. So here, that's where now we are going to talk of this molecule which we just said is what? Catabolite activator protein. This is one of those molecules which it's one of those molecules which activates the process of what? Transcription. Before maybe we talk of that, another good example here is going to be what? Lactose. We said that lactose is going to act as a positive inducer because when it is present, it's going to bind with what? With the operator, with the repressor there at the operator. Then the repressor is going to move out of the operator. So lactose is also one of the molecules which is controls the positive feedback mechanism of the Lac Operon. So now let's talk of the activators. Remember, we have activators and the inhibitors. Inhibitor, an example is the repressor. So examples of activators here, the molecules which can speed up this process. One of them is going to be that catabolite activator protein. So this, what happens here? What happens with this molecule? This one also will go back to the concentrations of glucose and what? And lactose. Glucose and lactose. So what happens is that if glucose is absent and lactose is present, we know that the Lac Operon is going to be turned on in this case. So what happens is that if glucose is present or is absent, there's going to be another molecule which is going to be activated within the cell. So there's going to be another molecule which is going to be activated. And this molecule is what we call cyclic adenosine monophosphate. So we have cyclic adenosine monophosphate. Okay, monophosphate. The concentration levels of this molecule depends on the concentration of glucose. if the concentration of glucose are low in the medium, the concentration of this cyclic adenosine monophosphate will increase. When the concentration of glucose are high in the medium, the concentration of this cyclic adenosine monophosphate will decrease. So these two, glucose and this molecule, they are inversely proportional to each other. And cyclic adenosine monophosphate is one of those activators which control positive feedback mechanism. So this is what we now want to see, how this cyclic adenosine monophosphate molecule is able to what? To activate this process of transcription in the Lac Operon. Okay, so what happens, we are saying that this, in the presence of lactose, the concentration in the medium of this molecule, so-called cyclic, by the way, this C is a lowercase, then the rest are uppercases, like that. That's how it is written. So we are saying that if glucose levels are low and we have lactose in the medium, the levels of cyclic adenosine monophosphate will increase in the medium. So this cyclic adenosine monophosphate has to bind where? At the cap site here. So this cyclic adenosine monophosphate does not have binding affinities of the cap site. So this means that this cyclic adenosine monophosphate has to combine with another molecule which can help it go and bind where? At the cap site. This is where now this molecule, catabolite activator protein, comes in. So we have high levels of cyclic adenosine monophosphate in the medium. This one has to combine with another molecule which can help it go and bind where? At the cap site there. So now, this is where now we have, this is going to be the, okay, this is the catabolite activator molecule. So this is the catabolite activator protein. Then what do we have now? We said we now have high concentration levels of CAMP, catabolite cyclic adenosine monophosphate. So this wants to go and bind at the cap site. So on its own, it cannot go and bind there. As a result, it has to combine with another molecule which can help it and go and bind where? At the cap site. So that molecule is what we call the catabolite activator molecule. So what will happen? These two are going to form this complex. So what we have here, what we have there, this is what? The CAMP. MP. So this now has formed a complex which we call CAMP complex. More like an enzyme and its substrate. So this is what is going to be formed. Once this is formed, then this CAMP complex is going to bind where? At the cap site. So this cap is just to help this CAMP to bind where? At the cap site. So we have that molecule here. Then this with its what? Cyclic adenosine monophosphate molecule. So when we have this one here now, this molecule is now going to trigger the binding of RNA polymerase at the promoter. This is going to be at the faster rate. The presence of this cyclic adenosine monophosphate at the cap site there is going to trigger the fast binding affinity of RNA polymerase at the promoter. As a result, this is going to facilitate the transcription of these genes at a fast. So that's how this becomes what? To be one of those molecules which control what? Positive mechanism, or the positive mechanism of the Lac Operon. Okay. So that's what happens. This is just an activator. This molecule is just an activator. It's going to facilitate the binding of RNA polymerase at the promoter. In this case, we have high levels of lactose, so we're expecting this repressor not to be there. It has to move out of there when it binds with the lactose. Then at a faster rate, this RNA polymerase is going to bind here because it will be with the hope of what? That molecule there. Then this RNA will transcribe these genes at a very fast rate. As a result, these genes are going to be produced at a faster rate, meaning that here the rate at which this lactose is going to be broken down in the cell of the bacteria is also going to increase. So going to be increase. So this is what we can talk about the positive control mechanism of the Lac Operon. Having reached this stage, then we can say we have reached the end of the Lac Operon. At least by now, you can go and answer questions on the Lac Operon. So thank you for watching this video. Keep looking forward for some more videos.